Well, it's right at noon, so I think we'll go ahead and get started. Just a couple kind of housekeeping notes we start on today. This is our third session of 4 that we scheduled here for November and December to give you an opportunity to hear from Montana experts about different aspects of coronavirus. We did record the previous 2, so if you'd like to go back and review those, they are posted on our website, the same website from which you logged into today's session. So, and just a reminder, the first one was by Dr. Andrea Marzi from Rocky Mountain Labs talking about how vaccines work. The second one in the series was from Dr. Mariana Carrera, a healthcare economist here at Montana State University, talking about the economics of subsidies, incentives, and various public policies. So those were the previous 2. They're available to you if you'd like to watch those at any time. As for today, there's a couple ways that you can interact with this. First of all, we have the question and answer pod at the bottom right there. And if you open that up, those questions will come just directly to me, and then no one else will see them. And I can ask those questions to our speaker as well. You're also welcome to use the chat window. If you select everyone on the webinar today will see who asked the question. So that's up to you. I'll monitor both of those. Feel free to send questions in throughout the presentation. If there's a nice break and we get a chance, we'll ask those during the presentation. Otherwise, we'll circle back to all of them at the end of the session today. So with that, let me introduce today's speaker. Today, we're fortunate to have Dr. Chris Nero with us. Dr. Nero works at Bozeman Health, which for those of you that aren't from Bozeman is our large primary hospital here in the Gallatin Valley. And specifically, Chris is a pathologist, so he's going to talk to us a lot about COVID in the laboratory. And just a side note about Chris, he also happens to be the 2020 winner of the Caregiver of the Year in Montana. So Chris, I'm going to turn it over to you, and thank you very much for taking time to join us today.
Sure. Thanks, Joel. And as Joel said, my name is Chris Nero. I'm a pathologist at Bozeman Health, also with Yellowstone Pathology based in Billings. And I'm going to give you a background of how I got into pathology and what pathologists do in general, because quite a few people don't realize all the different things we do in a hospital and in a healthcare setting. Some facts about viruses, and then we'll get into testing for COVID with PCR, antigen testing, and how to interpret COVID antibody testing. And then we'll get a little bit into the variants of the spike protein and how different variants affect how we test for those and look out for new variants that are occurring with SARS-CoV-2. So my road to pathology, I started off, I thought about medical school at a young age when I was in high school. My mom was a nurse, so I got a degree in chemistry, molecular biology at UConn, which is in Connecticut. And after that, I went to New York Medical College, which is based in New York City in Valhalla. And I had read books like medical detective books like this one below. And I thought that it'd be interesting to go into medicine and try to figure things out. I didn't quite know a lot about pathology at that point. But I ended up joining the Navy, and they paid for my medical school. And just to show my background, I did a year of surgery at Portsmouth Naval Hospital. You can see the image in the upper left. And I flew airplanes and helicopters in Pensacola, mainly as a way to get primer to learn what the pilots go through. And the Blue Angels, you can see up on the right, train out of Pensacola. And then this on the bottom right is a dunker that they put you in, which is kind of fun. They put you underwater and you have to swim out with blindfolds on. So I did some of the fun flight training stuff. And then I joined a squadron of Marines with F-18s out in South Carolina in Beaufort called the Checkerboards. And one course I got to take while I was up at USAMRIID in the Navy it's the Medical Management of Biological Casualties course. And this is a course that you can take as a flight doctor or a Marine flight surgeon. And I learned from Bill Patrick, who's in the lower left. He's the one who actually was involved in the 50s and 60s on looking at how anthrax could be used as a biological weapon. Our country shut down our biological weapon program in the 70s, but as you know, later on in 2000, there were the anthrax attacks, and he was called to be an expert witness on that. But I thought that was kind of interesting. And at that time is when I decided to go into pathology, because I kind of learned I was the expert in an area of teaching people about anthrax and smallpox. This was back in 2001. And I learned from a friend that pathologists, what they do is they look at puzzles and they figure them out. So I did pathology at Mass General Hospital in Boston. And this figure in the upper left is the first surgery under anesthesia that took place in the ether dome, and that took place at Mass General Hospital. So it's a hospital with a lot of history, and I saw quite a bit of pathology there. And afterwards, I decided as a fellowship after I finished my pathology training to go to Baylor College of Medicine in Houston, where I did molecular pathology. And this was a really good place to train because they had, there's Texas Children's Hospital, there's MD Anderson in the middle, and Baylor Genetics. So I got a lot of training in molecular pathology as a one-year fellowship after my pathology training. And molecular pathology, you can see in the upper left, you see the familiar double helix of DNA, deals with using molecular methods to help make diagnosis anywhere from pediatrics, newborn diseases to infectious diseases. And at the time, actually, I got training on designing tests for H1N1, which was going around in the early 2000s. So I joined Yellowstone Pathology after that in Billings. And there's a picture of me here in our lab in Billings, where we were one of the only labs in the whole region of Montana, Wyoming, Idaho to be doing molecular testing. And we were doing testing on tumors and some infectious disease testing. And so we were able to apply that. And then when COVID came around, we had some training and we had people ready to do testing. So what do pathologists do? There's a combination. We do anatomic pathology, which people may have heard about we will get tumor resections and we'll analyze those and tell what type of tumor. We'll look at biopsies of various body tissues to diagnose infectious diseases or inflammatory diseases. So that's the surgical pathology aspect. And then the clinical pathology part of pathology is managing the lab. And this encompasses microbiology, hematology, transfusion medicine, are some examples in a hospital. And we supervise the lab, make sure it's running correctly. And the other aspect is molecular pathology, which has become a bigger part of pathology in the last 10 to 20 years. And this is just an example of fluorescent in situ hybridization, where we use molecular probes to diagnose leukemias and lymphomas. So getting on to viruses. Viruses are all over throughout the environment. The various foods we eat will have different viruses, such as plant viruses, and they don't affect us. Most of these, and we have many viruses that live inside us, inside of the bacteria that are inside us, part of our microbiome. And what is a virus? A virus is an infectious obligate intracellular parasite with DNA or an RNA, and it's surrounded by a protein coat. So they're really not alive, but they kind of hijack our machinery of our cells to replicate. And viruses, as you can see here, are very small. They're a little larger, they're composed of proteins and amino and genetic material, but they're smaller than most of the known bacteria, although they found some larger viruses. And how many viruses could fit on the head of a pin? This is an example of a head of a pin, which actually has some bacteria on it, but 500 million rhinoviruses could fit on the head of a pin. So that's one reason it's so hard to avoid, the spread of virus infections, because there's so many of them, and one sneeze could, in theory, infect many, many people. How many viruses are inside our body? Well, quite a few of them live inside the bacteria in our body. There's actually 380 trillion viruses in our bodies, and most of those kind of live inside bacteria because we have 39 trillion bacteria and about equal number of human cells. It was once thought there were 10 times as many bacteria, but when further studies were done, we have about 30 trillion cells, the average adult, and about a little bit more of that in bacteria. And the bacteria can be in our gut, on our skin, in various other places in our mouth. And many of these are infected, per se, have viruses inside of them. And how far, if you took all a bacteriophage is one of these viruses that infects bacteria. And there's 10 to the 31 thought to be in the entire world. So 10 with 31 zeros. And the length is 120 nanometers. If we and I got this from some viral presentation, if you stretch these up back to back to back, how long would they stretch? Given that they're much smaller than a human cell. Well, there's so many of them that when it's multiplied out, it ends up being 10 to the 21 kilometers, which ends up being 10 to the 8th light years. So that could stretch to the next galaxy. So it shows how many viruses there are in the oceans all over the place that they would stretch very far. So how do we detect viruses? Since they are so small, we can't see them really with a microscope. We do a variety of different tests. We do antigen tests, which we'll talk about, molecular testing, and serology, which looks for the antibodies produced in response to the virus. The antigen test takes a sample of the fluid. Say if you swab somebody's nose, the fluid from that is placed on a sample pad, and then there's monoclonal antibodies which will bind that antigen, which is this reddish object, and then a signal probe will also bind the other side of the antigen, and that'll light up on this test strip. And if that's positive, then it indicates there's viral antigens present in that sample. Molecular tests, instead of looking at the proteins, which are the antigens, looks at the molecular material, the DNA or the RNA. So the first step in molecular test, you have to extract that DNA or RNA. And we'll talk about how that works. And then there would be molecular probes, which are in primers, and we'll talk more about those, which are complementary. They match up with those, and then they're detected on an instrument. So they're a little more complicated than antigen tests. And molecular methods, the advantage they have is they amplify the genetic material, meaning copy multiple times. And if you do 35 to 40 cycles, you can end up with billions and billions of copies. And this allows you to detect very small amounts of genetic material. And as far as antibody tests look for prior exposure to COVID or another SARS-CoV-2 or another virus. And these ones use the antigen to see if the person has previously generated antibodies to the virus. These can be induced either by having a prior infection or by vaccination. And if they're looking for the spike protein antibodies, they'll be positive in whether or not you've been either vaccinated or have had natural infection. When this outbreak occurred last March, there was no test developed for SARS-CoV-2 commercially. So we ended up teaming up with researchers from MSU, and this is Michelle Flanagan, and her lab at MSU had a lot of experience in developing molecular PCR tests. And they were able to help train our technical staff who hadn't done as much of this. And they followed the CDC protocol for SARS-CoV-2, and were able to help us get up and going with our lab in our hospital. So the SARS-CoV-2 genes, it has about 20 different genes, and some of those, the ones that we targeted, what you want to do is develop a primer sequence that will match up with that gene and not match up with any other viral genes. So what the process is to look at the whole genetic sequence, and we had the whole genetic sequence of SARS-CoV-2. It was 30,000 base pairs, as you can see here. And it had a variety of these genes. In the CDC assay, they had picked the N gene, the nucleocapsid gene, and developed a couple of primers for that. You could also target different areas of the viral genome, and it's kind of beneficial to target multiple areas to make sure that it's a true positive, and in case a mutation happens that would block the production of a PCR product in one area of the genome, you can still pick it up in another area. So at first you develop it, it's called in silico, use a computer program to develop those primers, and then you test it out in the wet lab on the laboratory level. So the CDC had developed these primers and probes, and then we had to test them along with Michelle Flanagan and to see if they worked in the wet lab setting. So these PCR tests are designed to be highly specific for SARS-CoV-2. They do not cross-react with other viruses. So if you put them through a database computer program, it would make sure that they don't interact with other coronaviruses or any other virus. So the first instrument we used in our PCR assay was a Biorad. We used Biorad instruments that were provided by MSU Bozeman, and it was a long process. We had to do, Michelle helped get it validated for us, but the whole testing process takes about 5 hours. So there's a lot of manual steps of extracting the RNA, followed by PCR, and a lot of manual pipetting, taking about 5 hours start to finish. And with this PCR machine, you go through the PCR cycles, which is you heat it up. It's kind of a cycle of heating and cooling. And when you heat, the primers and probes separate. When you cool down, they come back together. And then you get an amplification. And the PCR product will develop after about 20 to 30 cycles. And when you develop these cycles, you can actually come up with a standard curve. And as we said before, with exponential amplification of the target genes, meaning you're copying it multiple times, the tests are highly sensitive. You can copy a very small amount, and in this assay, you can get down to the nanogram levels of starting product and be able to amplify that. And that gives you an idea of the viral load, which is another term, the amount of virus in the sample by when it amplifies. If it amplifies early, like at 10 to 15 cycles, then there's a lot more viral product than if it's at 30 cycles. After that, so we used that assay for several months, and it worked, but we had a lower volume. We couldn't quite handle the volume, so we ended up buying some more instruments. One of them is a Kingfisher automated extractor, and this allowed us to extract the RNA from multiple samples at once. You could load up to 96 samples and automate that part of it. We still needed to do manual pipetting for the PCR reaction, but this gave us a higher throughput. We were able to get up from initially about 80 samples a day. With this assay, we get up to 300 to 400 samples, could be run over 8 to 10 hours. And there was a little bit less risk of carryover or contamination due to less pipetting. The one issue with PCR being so sensitive, if you pipette and you have a little micro droplet that's carried over, it can actually cause a false positive in another sample. So we ended up obtaining 2 PCR machines, replacing the Biorad. These were ABI 7500 FastDX PCR machines. So we could do extractions and then run the pipette and then run the PCR on one machine and then do another set of extractions and run them on the other. Our rates of carryover went down after this, and carryover is when you have a micro droplet. Even a minuscule amount of virus can show up in another sample. So we wanted to eliminate that. Another assay that we've had before is the BioFire, and this is considered a rapid assay, whereas that previous PCR assay takes about 3 to 4 hours to run. This is a rapid assay by BioFire. It takes about 45 minutes to an hour to run. And we had gotten a limited supply of these early on in the pandemic, but given that everyone else wanted to also do testing, BioFire gave us limited allocation. And this one uses nested PCR. So there's a PCR reaction where the primers and probes match up, and then it's amplified again in a second reaction. So it's really highly specific with those several reactions. And BioFire can also run testing multiple different viruses and bacteria at the same time. So we have this panel running currently, and we can detect up to 20 different viruses and bacteria in about 45 minutes. And these are primarily used for critically ill patients when they really want to know quickly which bacteria or virus is causing the infection. Another assay we had as a rapid molecular test was the Abbott ID Now, and it uses isothermal amplification. So instead of heating up and cooling down, it's all at the same temperature, and the primers bind and they form these loops, and then you get amplification. And this was definitely helpful throughout our early times of the pandemic to have rapid tests so we could quickly tell the doctors and surgeons whether somebody was infected either prior going to surgery or admitting to the hospital. And this has also higher sensitivity than antigen testing. And we ran the ABI 7500 for about 7 or 8 months. And then in the summer of this summer, we were able to get reagents finally for the hologic Panther, which was an instrument we already had to run various testing on women's health tests, such as pap smears, such as human papillomavirus testing. And we had this instrument, but we couldn't get reagents early on. But finally, we got reagents this summer. And because the whole process is automated with extraction and PCR and amplification, this one had less risk of carryover and risk of contamination. So we stopped doing manual PCR in the summer, and we just ran the Panther. And another interesting method of testing is not just doing nasal swab, which I was talking about previously, is doing saliva-based testing. And this was used in the NBA. It was either easier sample collection for the basketball players, and in other settings it's been used. And in this particular test, they did it without an extraction, which lowered the sensitivity a little bit, but it was still enough to successfully detect the virus. And both nasopharyngeal and saliva samples, you can see the green and the blue, showed similar rates of detection. And PCR being so sensitive, it can even pick up people well after they're out of the infectious period, which is thought to be the first 10 days of the infection, as we can see here, the red and the green. And just to give an idea of how long these tests are positive, the blue line is the PCR test. So the PCR test can be sensitive up to several days before symptoms appear. And then once symptoms appear in this black dotted line, PCR will remain positive in most patients. You can see the load here, out to 4 to 5 weeks. So the one thing not to do is not to test for cure. They're not designed to see if you're no longer infectious. It's really clinically based. The first 10 days in most patients, that's your infectious period, as long as you're getting better. But the PCR can be positive out to 6 to 8 weeks because it can pick up remnants of the viral genetic material. Antigen testing is primarily positive in the first week or so, and then it goes negative, so it's less sensitive. And then we'll talk about antibodies. They tend to be positive after the first 10 days and remain positive for a longer period of time. So this is just another diagram that shows you the antigen test is positive in the first week to 10 days. So it does more correlate with that infectious period. And then the PCR can remain positive for quite a long period. Antibodies will remain positive 6 months or more following an infection. In different testing strategies that are used, you can have, in the hospital setting, it's diagnostic. We're looking to see if somebody is symptomatic or high risk exposures, and you can do antigen testing or PCR. In a population level, there's surveillance testing, and this has been pushed to do rapid antigen testing, either in schools or PCR in a variety of settings, and such as nursing homes, to make sure we detect them early before people actually have symptoms. And moving on to the spike protein, as we know, the spike protein is where coronavirus kind of gets its name, as it's got spikes coming off of the virus. And that's the primary point of entry for the virus into the cells. So it has this receptor binding domain where it binds to the cell. And this is where a lot of those mutations happen that allow it to get into the cell. And this is an image of showing how the virus actually gets into the cell, that the spike protein interacts with cells in our respiratory tract, such as ACE2 and this TMPRSS2. And there's a cleavage of the protein on the virus, which allows it to fuse to our cell and join in. So those amino acids, if they're altered, it can allow the virus to enter more rapidly into the human cell and have a higher level of infectivity, such as we have seen with the Delta virus variant. So the antibodies that our body develops, many of them are targeting these spike proteins. And these are also, the vaccines also elicit antibodies to these spike proteins. So it's, as we're looking at these new variants that are occurring, such as Omicron and the Delta variant, we're going to look at those areas because that's where the virus is going to be changing to try to, potentially become more infectious as it mutates here in these various areas on the spike protein, if any of these give it a selective advantage in infecting human cells, it'll quickly become the predominant virus in a community. And we saw this with the Delta variant over the summer, that once it entered a community, it quickly overwhelmed all the other variants and was the predominant one. And we'll have to see what happens with the Omicron variant if it has a selective advantage. So as far as our lab, we don't do variant testing, but this is done. MSU Bozeman has done it previously. I know they were looking for the Delta variant and the other Beta variant, but various academic centers do this, and it's thought that the more often we do test the variants, we can pick up them as they occur and be ready for any new treatments that are required or any other situations. So summing it up, pathology is a branch of medicine that uses laboratory methods to diagnose disease. And we have to be able to get fast and precise diagnoses to the doctors. We're kind of considered the consultant for the doctors to let them know quickly so they can treat their patients. And this was definitely true with COVID. They needed rapid diagnosis. So we were fortunate to partner with MSU Bozeman to develop COVID testing so we could get it done very quickly. And COVID can be diagnosed either using antigen or PCR tests. So they have antigen has the benefit of just being positive mainly during that infectious period, but it is less sensitive. And PCR tests are more sensitive but can be positive for a long period after the infection when people are no longer infectious. So prior disease can be diagnosed with the serology tests such as the antibody tests. However, one thing to remember is the serology tests cannot be used to show immunity to COVID. We haven't quite got to that level. Hopefully, they'll have some standardization of the antibody test to see what level of the antibodies is required to show immunity to SARS-CoV-2. And I'll be happy to take any questions.
All right, well, thank you, Dr. Nero. We've had 4 questions that have come in so far, but please continue to send them in the question and answer pod or the chat window, and we'll kind of work through those. So the first one, I'm an economist, so I picked this one to go first. How expensive was that lab equipment that you had to acquire in the last year to handle those tests?
Yeah, the various equipment, they range. The PCR machines are about, I think, 50,000 a piece, and then the extractor, and I try to remember the numbers. I think it cost us close to 200,000 to set up that equipment. The reagents are also quite expensive. The Panther machine, I know, is about in the $150,000 range, and the reagents tend to be expensive also. We also do some testing in Billings, and the machine we got there was a little bit cheaper on that end. It was a $25,000, but they're in that range, any for $25,000 to $50,000. And I know the testing costs are somewhere. You have your labor costs plus your reagents, and they're anywhere from $50 to $100. And I think the going rate for a COVID test is about $100 for most labs.
All right, thank you. Next one, and I think you did mention this topic just briefly, but somebody wanted to know, why do we test wastewater, and can you tell us more about that process and how it's helpful?
Yeah, if there is a period where, we can diagnose people in the hospital, but by the time they present to the hospital, there's probably already a major outbreak going on. The wastewater can be an early indicator. And I know they use this in the summer in periods where we didn't have many cases. If you start seeing the viral levels, it's kind of, they can determine it quantitatively go up in the wastewater. It's kind of an early predictor that there'll be an outbreak of COVID. And you could use this for other viral infections also by the quantity of the virus, because it does come out in various body secretions or liquids. And the overall amount can be a good kind of sampling measure of the environment, before people actually present to the hospital with symptomatic disease.
All right. Next up, does Montana have the same testing abilities as larger hospitals in more metropolitan areas?
Yes, those tests that we're using, such as the Panther, early on, I think because our population was smaller, we weren't being, and we had less of an outbreak early on in March. It was really being the national lab testing companies such as Hologic, who provided us with the Panther, weren't allocating us tests. So early on, we didn't have it, but we were fortunate that we were able to do those home developed kind of tests, laboratory developed tests as we describe them with MSU. And we were the first lab, I think, definitely in the state to develop our own assay here and be running it with, because we had the partnership with MSU. So at that time, we had the same tests as everybody else. And currently we're actually running the Hologic Panther is probably one of the most commonly used tests in the country to test for SARS-CoV-2. So it's in the rapid tests we have, such as the BioFire, are also the same tests that most of the other labs throughout the country use, most of the other hospitals.
All right, thanks. Next up here, and I'm going to read this one because I might need more academic background just to understand the question. But what do you know about the T-Detect test that was approved by FDA to detect T cell immunity levels to COVID? Why can't serologic tests be used to show immunity? And how do we prove current immunity?
Yeah, that I don't know. I've heard of tests like that test. I don't know that particular test, but it is there is a component of immunity, and this is kind of what we're learning about. The easier measure is serologic assays, but as we know, the antibodies are produced by your B cells, and this person is referring to the T cell component of immunity, and that is used in some other disorders such as tuberculosis to check for immunity. But I think there's not quite in order to have tests applied, they have to go through the FDA authorization approval process in order for them to be commercially available. And I think at this point, they haven't met that level of commercial, up to the level of development that the FDA has approved them. I know for serological assays, there's a company or two that has tried to claim that their particular antibody assay can prove immunity, but the FDA has put out warnings recently that you cannot use any test to show immunity. It just hasn't gotten to that level where we do have that ability for like measles, for diseases that have been around for a while. They eventually figured out what level and what tests can show that somebody's immune. But I think with COVID and SARS-CoV-2, the fact that it seems to be having various mutations and variants, it might be a little bit hard to say how effective, what is an appropriate antibody level, given that there might be a new variant showing up. So it may take a little bit longer, and all of us want that to come quickly, but we're just not at that level yet that the FDA has approved that kind of assay. Because our hospital, typically, all of our assays have to be FDA authorized or approved, and that goes for the hospitals throughout the country, because we want tests that are reliable and accurate. We don't want unreliable testing to occur.
All right, thank you for that one. Here's another one that I'm glad we got because this was some water cooler discussion at my office here recently. It says, I recently got a blood draw and a panel of results. I've had the vaccine and it gave me an IgG quant result.
Okay.
And then it said, these results are not to be interpreted as a degree of immunity. What do those results mean to that person?
Yeah, our instrument also we have a method that measures spike protein antibody levels, and we don't report out the levels because it's unknown what that number means. In general, what you would want is to show that this antibody, and this is what would have to be proved to the FDA, it actually neutralizes the spike, the coronavirus in a test tube environment. And if you can show that that antibody actually neutralizes the virus, and then your test actually tests for that antibody, then the higher the level, the better. It would be better if whatever your cutoff is for immunity. If immunity says 100, if your level is 500, then you're more certain that you're going to be immune than if you're down either at or below the cutoff. So right now, that number, we really can't do a lot with that. It's maybe nice to know it's there. I mean, but they're not even actually really recommending we check antibody levels because we don't really know what to do with the information. So you don't want to get false reassurance that you can take off your mask and do a variety of things. We really still need to be careful until we get enough data on what antibody levels and how we measure them can show that we're actually protected.
All right, thank you. So there's two questions. I'm going to merge into one here for you. Both are around at-home COVID tests. So one, do you have any recommendations about whether or not people should use them? And two, how do you recommend people use those versus coming in and getting a formal lab test?
Yeah, there are a variety. It'd be hard to comment on all the at-home tests because they can either be antigen tests or they can be, well, the PCR tests, generally you have to sample them and send them into a lab. If you're doing them at home, they're probably an antigen test, which is kind of like a pregnancy test that you can do at home. Those could be useful. I mean, if you have an antigen test, if somebody becomes symptomatic and they want to test with the antigen and it ends up being positive and it's run correctly, that can be taken as evidence that they do have SARS-CoV-2 COVID infection. If it's negative and you're symptomatic, because the antigen's a little less sensitive, you might still want to go in, especially if you're having any higher risk exposures to other people, and get tested by PCR, because it's not, and I think that's generally recommended by the FDA, that not to completely rely on antigen testing, that if you actually become symptomatic, go in and get a PCR test at a hospital, because the assays are, they're run, they have quality control and highly sensitive assays that are much more reliable than a home antigen type test. But it could be used in some environments, and this is what Michael Minna he's one of the advocates of doing a lot of screening surveillance testing with antigen testing, because then you can pick up somebody, and if they do end up being positive, they could confirm it with a PCR assay, and before they go on to expose anybody else or get rapid treatment. So it really depends on what the home assay is, but they can be used, and I'm just not, there's a lot of different names out there, a lot of different companies, and I know some of them market them as PCR tests where you swab and then you send it off. But I think most people are probably better off just going to the tested at a hospital type setting or a reputable lab that has a good PCR test.
All right. Next one here is the news reports ICU and hospital bed capacity, and sometimes our hospitals are full. Are the labs also at their capacity?
Yeah, there were periods of time, especially early on, where we only had that Biorad assay that we didn't quite, we could test about 80 a day. This was back a year ago, November, and we had to send our extra tests to the state lab. And those sometimes took about a week to come back, and that was kind of too long. So that's why we moved on to the other instruments. And since we got the ABI TACPath assay and the Panther, we've been able to handle the volume. So with the Panther assay, we've been able to we can run up to 500 tests a day. And currently, thankfully, with the numbers coming down, we're not running that many tests now. We were up to about 400 to 500 a day, and that's about our capacity. But now we're a little less than that. We're probably half that now. So because of those automated instruments, we're doing much better. And the lab component hasn't been a limiting factor, at least since in 2021. Back in 2020, it was somewhat of a limiting factor, but we've been able to get all the tests essentially out in 24 to 48 hours for people in the Gallatin County.
All right, well, we've gotten through all the participant questions, so I'm going to ask you one more, and then any final comments you want to add to that, feel free to go with. What are you expecting or looking forward to in the next 3 to 6 months in terms of new tests or equipment or things that might be on the horizon with respect to your lab and kind of managing coronavirus?
Yeah, with the current instrument we have and the fact that the Panther assay, it detects multiple targets on SARS-CoV-2. One thing always to look for is if there's a new variant that somehow escapes detection because it has a mutation in that area of the genome that you're targeting. And we've paid attention to that, and we talked to the companies that market these tests, and that's something always to be aware of. But currently, the mutations that are occurring are mainly in the spike protein, and our assay detects different regions of the genome of the virus. So we haven't had any issues with missing the or having false negatives for SARS-CoV-2. So we're with the hologic Panther assay, I think we're doing pretty well. The one thing to look forward to is the antibody assays and serologic tests. And I'd like to look more into seeing when somebody can actually say there's some immunity, what level is required for immunity, because that would help people who've either been vaccinated or had natural exposures to know what level they're at and if they have some level of protection. And I think hopefully that's going to come out in the next several months. And at some point, viruses like SARS-CoV-2, they're going to have variants that come up, but it'd be nice to know what level is immunity. Then you have a little bit more peace of mind about not worrying about the next new variant that comes out. But that's kind of to be determined as things occur. We have to kind of look at these, and especially until a larger percentage of the world's population is vaccinated, we're likely to see new variants occurring.
All right, well, thank you very much, Dr. Nero. I learned a lot today, and thank you everybody for sending in the excellent questions. And again, for those of you that have been following the whole series, 2 weeks from today, we will have our fourth session, our final one before the end of the year, with Dr. Sophia Newcomer from the University of Montana will be presenting on that section. So thank you again, Dr. Nero, and thank you everybody for joining us today, and have a great rest of your day.
Okay, thanks a lot, Joel.